Genetics and Genomics in Medicine, Chapter 2, Fundamentals of Gene Structure, Gene Expression, and Human Genome Organization. Module 2, RNA Genes and Non-Coding RNA. RNA genes do not make polypeptides. Instead, they make a functional RNA product. They are often abbreviated as NC for non-coding NC RNA. Like proteins and mRNA, non-coding RNAs are made as precursors that often undergo enzymatic cleavage to become mature gene expression products. They are also subject to chemical modification. Minority bases such as dihydrouridine or pseudouridine and various methylated bases are quite common. Until recently, non-coding RNAs were largely viewed as having important but rather dull functions. For the most part, they seem to act as ubiquitous accessory molecules that work directly or indirectly in protein production. After ribosomal and transfer RNAs, we came to know about various other ubiquitous small nuclear RNAs, snRNAs, and small nucleolar snORNAs that chemically modify specific bases in RNA. Small caudal body RNAs, scRNAs that chemically modify spliceosomal RNA, and certain RNA enzymes known as ribozymes that cleave tRNA and RNA precursors. All of these types of RNA can be viewed as general accessory molecules that, like ribosomal RNA and tRNA, are ultimately engaged in supporting protein synthesis. In stark contrast to RNA, proteins were viewed as the functionally important endpoints of genetic information, the exciting pace setters that performed myriad rows in cells. Today, non-coding RNAs are recognized for their regulatory roles in gene expression, as well as the fact that mutations in non-coding RNAs are often implicated in genetic disorders and cancer. In fact, today, RNA itself is being targeted as a therapeutic option for new approaches to treating disease. This slide shows the highly developed secondary structure found in RNA. Unlike DNA, which has a stiff, rod-like structure, cellular RNAs are very flexible. Being single-stranded, they have a high degree of secondary structure that can be caused by interchain hydrogen bonding that can lead to a variety of highly and intricately folded structures, as evident in the ribosomal RNA shown in this slide. The 5' ends of the RNA are highlighted for clarity. The basic motifs are the stem loop structures. The stem loop structure, which is formed when RNA folds back on itself, such that two short regions can base pair to form the stem, while intervening sequences loop out. Note that GU base pairs form an RNA in addition to GC and AU base pairs. This slide demonstrates the versatility of non-coding RNA. The two panels on the leftmost side show ubiquitously expressed RNAs that are important in generally assisting protein production and export, including RNA families that supervise the maturation of other RNAs. The central panel includes RNAs involved in DNA replication. The ribosome RNase MRP has a crucial role in initiating mitochondrial DNA replication, as well as cleaving pre-transfer RNA and developmentally regulated telomerase regulators. TERC is the RNA component of telomerase. TERA is telomere RNA. Diverse classes of RNA regulate gene expression. In addition to ubiquitous RNAs, such as the listed RNA that have the general roles in transcription, many classes of RNA regulate specific target genes and are typically restricted in expression. They work at different levels: transcription, such as antisense RNAs, splicing and translation, specifically miRNA, which binds to certain regulatory sequences in the untranslated regions of the target mRNAs. Some RNAs, notably the highly prevalent classes of circular RNAs, regulate the interaction between miRNA and their targets, piRNAs, and to a smaller extent, endogenous short interfering RNAs, endo-siRNA, are responsible for silencing transposable elements in germline cells. RNA as specific regulators. The first examples of more specific regulatory RNAs were discovered more than 20 years ago, and for a long time they were considered to be interesting but exceptional cases. They included RNAs that work in epigenetic regulation to produce monoallelic gene expression, where it is normal that only one of the two parental alleles is expressed. Although the Human Genome Project has been crucial in an important identification of novel protein coding genes, it has not been so effective at identifying RNA genes. However, subsequent studies of genome-wide transcription, plus cross-genome comparisons and bioinformatic analyses, revealed many thousands of novel RNA genes. Many of these genes make regulatory non-coding RNAs that are expressed in certain cell types only, including large families of long non-coding RNAs, tiny non-coding RNAs, and circular RNAs. Long non-coding RNAs. Long regulatory non-coding RNAs come in two broad classes: antisense RNA, which are transcribed using the sense strand of a gene as the template and are not subject to cleavage in RNA splicing. As a result, they can be quite large, often many thousands of nucleotides long. They work by binding to the complementary sense RNA produced from the gene, downregulating gene expression. A second class of long regulatory RNAs are formed from primary transcripts and are typically processed like the primary transcripts of protein coding genes. Many of these RNAs regulate neighboring genes, but some control expression of genes on other chromosomes. Tiny non-coding RNA. These RNA molecules are very small, typically less than 35 nucleotides long, and function in human cells. They include many cytoplasmic microRNAs that act at different stages of cellular development. They are important in a wide variety of cellular processes. Human germ cells also make many thousands of different 26 to 32 nucleotide PIWI, or protein interacting RNAs. These PIRNAs work in germ cells to damp down excess activity of transposons, or mobile genetic elements. Active mobile genetic elements in a genome can make a copy that migrates to a new location in our genome and can be harmful by disrupting genes or inappropriately activating some types of cancer genes. So they play a very important role. And finally, there are competing endogenous RNAs, or pseudogenes. Certain regulatory RNAs that contain multiple miRNA binding sequences work by competing with an mRNA for access to its miRNA regulators. They include RNA transcripts made by some defective copies of protein genes called pseudogenes. Most of the circular RNAs overlap with protein coding genes and arise by circularized splicing.